The topic of the present presentation is phytoplankton, minuscule entities with immense importance in aquatic ecosystems. Myself, Obhik Kumar Chaudhuri, Assistant Professor at Department of Botany, Ramakrishna Mission, Vivekananda Centenary College, will be presenting this work. To begin with, what are plankton? The word plankton has been derived from the Greek word planktons, which means to wander. Now, these are basically aggregates or collection of weakly swimming or drifting organisms. Now, talking about swimming or drifting organism, phytoplankton or plankton can be categorized as swimmers or drifters. Those which have an organ of motility, they can move along the direction of current or against the direction of current using their organs of motility as per their need for food and feed. However, those plankton which do not have an organ of motility only drifts along the direction of current in lotic ecosystem. Those forms are called the drifters and not the swimmers. Now, plankton can be both microscopic or macroscopic in size. Few phytoplankton are macroscopic, but majority of zooplankton are macroscopic. So obviously, the plant forms are called phytoplankton and the animal forms are called zooplankton. Phytoplankton encompasses the photosynthetic prokaryotes and the photosynthetic protists. So, based upon the nature of plankton, they can be categorized as holoplankton and meroplankton. Holoplankton are those planktons which remain planktonic all throughout their life cycle. One example is diatom. On the other hand, meroplankton are those plankton which remain planktonic up to a certain stage of their life cycle and later on they become non-planktonic. One classical example is the genus Rhizoclonium belonging to the order Cladophorales. Now, in the juvenile form of Rhizoclonium, they do not have the rhizoidal branch and at that stage they remain as drifters or planktonic. But as they mature, they develop this rhizoidal branch by which they start attaching to the substratum. At this stage, they do not remain planktonic and hence, since they remain planktonic up to a certain stage of life and later on become non-planktonic, Rhizoclonium is considered as a meroplankton. Now, based upon the ecologically distinct groups that are present as phytoplankton in aquatic ecosystem, we can observe that diatoms are the largest group of phytoplankton. They belong to the class Bacillariophyceae under group 4 as per Lee's classification. The second most abundant group is the dinoflagellates, which belong to group 3 as per Lee classification. Then there are a host of other members of Heterocontophyta, that includes the haptophytes, members of Primnosiophyta, which are also known as microflagellates. Then there are some green algal forms like Cnidasmus, which can be planktonic, or Pediastrum, which can also be planktonic, and they belong to group 2 as per Leake's classification. And finally there are cyanobacteria, which belong to group 1. So here we can see that all the different groups of algae can constitute phytoplankton and it is not restricted to any particular group or any particular class. So these are some of the images of the major diatom groups that are observed in natural lotic system. So there are diatoms, then there is cyanobacteria like Microcystis aeruginosa, then there are several different types of dinoflagellates, and then there are green algal forms as well like Pediastrum and so on, like members of Cnidasmus, And then there are haptophytes like coccolithophores that constitute majority of phytoplankton populations. Now, if we like to classify phytoplankton based upon their sizes, so broadly 4 different size classes are there. Now, this classification was initially proposed by Seibert, and it has also been adopted in some book by the present authors. Now, there are larger forms like a megaplankton or an ultraplankton, but those are mostly zooplankton. Among phytoplankton, the largest forms are the microplankton that ranges in size from 20 to 200 micron. These are the phytoplankton that are observable and can be identified under the oil immersion objective of a compound microscope. Then we have nanoplankton, which ranges in size from 2 to 20 micron, and this includes taxa like Nanochloropsis, Ostriococcus, and so on, which are very small in size and often difficult to observe under a oil immersion objective of a compound microscope. Then we have further smaller planktons known as picoplankton that ranges in size from 0.2 to 2 micron. These include the cyanobacteria like Cyanococcus, Cyanocystis, and so on. Lastly, there are also planktonic forms which are less than 0.2 micron in size, and they are regarded as the femtoplankton. This mostly includes the cyanophages. Now, in this plate, we can see that the top panel represents the centric forms of diatoms, which includes some filamentous types or rather colonial forms also, which adhere with each other and form these chains, and that is also observed in case of the pinnate forms, which come together and form this kind of aggregates. So these are unicells that come together and they can form chains. Then these are the dinoflagellates. Now, as we can see that in case of dinoflagellate, the position of flagella is an important property. Either there can be a posterior flagella and a transverse flagella, and that kind of dinoflagellate are called the dinocont, whereas both flagella can emerge from the apical end, and that kind of dinoflagellates are called the desmoconts. Now, as we can see that in taxa like Noctiluca scintillans, Lingulodinium, Polyhedrum, and in Pyrocystis, they give this brilliant bluish color under a dark background. So this phenomena is called bioluminescence. This we will be discussing in our coming slide. Now here are two contrasting pictures of bloom phenomena caused by different phytoplankton taxa. One is caused by the genus Phyocystis poucheti near the North Sea, and it forms a dull looking foamy bloom. On the other hand, we see that there is a bloom by Emiliania Huxley, and it gives a brilliant turquoise blue milky color to the sea. So there can be different genera, and the bloom of these genera can be very different morphologically or from the external appearance. And then we have the prokaryotic phytoplankton that includes members like Prochlorococcus, which are very small in size and can be very abundant as well. They are mostly present at temperature range of 20 to 30 degrees centigrade. So they mostly occupy the tropical region. Whereas there are other members like Cyanococcus, which are also very small in size, and they can live in temperature of above 5 degrees centigrade. And they are very abundant in the euphotic zone of lotic systems. Now, why phytoplankton are important? What is their importance? Now, obviously, since they are photosynthetic, so obviously as a byproduct of photosynthesis, they releases oxygen, and that makes up more than 50% of the global oxygen concentration. So that way phytoplankton is a very important constituent. The other thing is that since it is photosynthetic, so obviously they sequester some amount of inorganic carbon dioxide from the atmosphere. So in that way also they are minimizing the high carbon concentration in the atmosphere around us. So in one hand it is producing oxygen for our benefit, on the other hand it is sequestering carbon dioxide to make the environment clean. So in both ways phytoplanktons are very important. The other important aspect is that since they are the producers of aquatic ecosystems, so obviously they regulate the functioning of the pelagic food web. So more the carbon uptake, better the efficiency of phytoplankton, greater will be the energy input in pelagic food web. Moving on, one another important aspect regarding phytoplankton is the biological pump. Now obviously the carbon dioxide is produced in air. Now since the specific gravity of seawater is high, so it allows greater solubility of carbon dioxide in marine water as compared to oxygen. So, and as temperature is rising, so the water mass increases in volume, and so it can take up more and more carbon dioxide into the system in the form of dissolved inorganic carbon. Now once within the water column, the phytoplanktons take up this dissolved inorganic carbon either in its gaseous state or in the form of bicarbonate and convert them into dissolved organic carbon. Now life forms in aquatic ecosystem can utilize organic carbon, but no life form other than phytoplankton or plant system can utilize dissolved inorganic carbon. So this conversion of inorganic carbon to organic carbon is an important step in an aquatic ecosystem. Subsequently, these DOC produced by phytoplankton are taken up by the zooplanktons and they form the particulate organic carbon. Now this particulate organic carbon are discharged in the form of fecal particle matter or by egestion. They are again recycled by the zooplanktons. The POC or the PIC that are produced are either recycled by the zooplanktons and again they contribute to the POC or PIC, or they are buried in the bottom of the sea where they are acted upon by the bacterial population. Now this bacterial population undergoes breakdown of this POC and releases carbon dioxide, which is again cycled back to the surface of the earth. So by this way, phytoplanktons actually participate in the global carbon cycle. The other important aspect is that as carbon dioxide enters into the water column, so it forms bicarbonate and proton by forming carbonic acid which dissociates into proton and bicarbonate. This bicarbonate is subsequently broken down into form carbonate. Now obviously both carbonate and bicarbonate are present in the aquatic ecosystem. Now as more and more carbon dioxide enters the aquatic ecosystem, so the amount of bicarbonate increases in this system, which changes the ratio of bicarbonate is to carbonate in the aquatic ecosystem. Now carbonate is an assimilatable form, but bicarbonate is not an assimilatable form. So those taxa like an Emilinia Huxley which assimilate calcium carbonate needs this carbonate. However, as bicarbonate concentration keeps on increasing due to ocean acidification and increased solubilization of carbon dioxide, so the carbonate concentration drops and the availability of carbonate becomes less and less, and the concentration of bicarbonate become more and more in the aquatic ecosystem. So this makes assimilation of carbonate in the form of calcium carbonate by this coccolithophores a difficult step. So this decrease in pH results in less availability of carbonate, thereby making the shells of these coccolithophores more brittle. That in turn will hamper the ecosystem functioning. So this upper panel image clearly shows that how the coccolithophore responds to environmental change and their shell formation. The other important aspect regarding phytoplankton is the possibility of using them as a proxy to understand the trophic status of the habitat. Now, there are two terms that interest us. One is water quality and the other is trophic status. Now, often we tend to mix these two terms, but water quality refers to the standard of water with respect to human use, whereas trophic status refers to the water condition with respect to the ecosystem functioning. So even though the status of a water with respect to quality can be good, it may not be good with respect to the trophic state. So in different countries under European Union, the Marine Framework Strategic Directive or MFSD has proposed phytoplankton as an important proxy for monitoring of aquatic ecosystem. Based upon that, from a long time, several indices has been proposed that utilizes data from phytoplankton populations to predict the health of aquatic ecosystem. Some of them are like the Niger's algal index. Then there is this PhycoI index, that is phytoplankton community index. Then in further down we can see that Palmer's algal pollution index was also proposed. Then in recent times other indexes like the trophic diatom index has been proposed. All of these are utilized to understand the trophic state of the habitat, whether it is eutrophic or oligotrophic or mesotrophic, whatever the condition is, and how it benefits or hampers the functioning of the ecosystem. That is looked through using phytoplankton as a proxy. Moving on, the other interesting aspect regarding phytoplankton is the property of bioluminescence. Now in this given image we can see that all different parts of Australia or Hong Kong or Belgium and in places like Mosquito Bay in Peru, all of these places show this phenomena of bioluminescence. Now in places like the Mosquito Bay, even recreational tourist activity are also there, where this bioluminescence event is considered to be an annual event for the tourists. So interestingly, the process of bioluminescence is restricted only to marine ecosystems or marine systems. Even though there can be phytoplankton in other systems, but there it does not show bioluminescence. One important reason can be that the mechanical shear stress caused due to wave action can be an important stimulant for the process of bioluminescence. Now, if we look precisely into the process of bioluminescence, we can see that within the tonoplast or within the vacuole surrounded by the tonoplast membrane, there are specialized structures called scintillons, which houses a system of luciferin, luciferase and luciferin binding protein together that actually functions in bioluminescence. Now, luciferin is actually a substrate for the enzyme luciferase. Now, when luciferin, luciferase and luciferin binding protein remains together, it is an inactive complex, and in that situation luciferin does not come in contact with luciferase, and as a result of which no bioluminescence occurs. But as proton concentration increases and there is a decrease in the pH, so luciferin, oxyluciferin and LBP dissociates from each other, and luciferin binds to luciferase and gets converted to oxyluciferin with the emission of a beautiful bluish green light. Now this light emission is basically the process of bioluminescence. Now It is obviously a pH dependent process, so the induction of acidic pH or the increase in proton concentration within the tonoplast membrane and the regulation of the same is an important factor to drive the process of bioluminescence. Now, this tonoplast membrane depolarization is again dependent on mechanical shear stress. So, since wave action causes a more or higher level of mechanical shear stress, so that is possibly the reason because of which bioluminescence phenomena is observed only in marine systems and not in other aquatic ecosystems. So moving on, the next aspect that we need to put some light on is the factors that regulate phytoplankton populations. So there are broadly 4 basic factors, among which the foremost is the light. Now obviously, since phytoplankton are photosynthetic organisms, they require light. Now if we classify the photic zone or the light available zone, then there is the uppermost layer is called the epilimnion, the intermediate layer is called the hypolimnion, and in between there is this metalimnion layer. Now the epilimnion receives the lower wavelengths of light having the higher energy content. However, photosynthesis happens in the red wavelength of light at lower energy. Now, in the metalimnion, even though the higher red wavelength of light reaches, the lower wavelengths of light does not reach the metalimnion. So, in one way, the phytoplankton can prevent itself from chlorophyll bleaching or photobleaching by avoiding these lower wavelengths of light by coming into the metalimnion. On the other hand, it can receive the higher red wavelength of light having lower energy and undergo photosynthesis. So, the metalimnion is the preferred zone for the growth and development of phytoplankton population. That is why the metalimnion zone is also regarded as the zone of chlorophyll maxima. The next important aspect is the availability of nutrients. Now, nutrients can be both with respect to nitrogen and phosphorus. These are the 2 principal nutrients that are required by any living population, and phytoplankton are no exception. So, nitrogen can be availed in the form of inorganic nitrogen, in the form of nitrate, nitrite, or ammonia, or as a summation of all this, it is said as dissolved inorganic nitrogen or DIN. Now, under optimum availability of nitrogen, obviously diverse phytoplankton groups can grow and it will support different kinds of phytoplankton. However, when nitrogen availability becomes limited, so in that situation, only the efficient nitrogen utilizers can utilize the lower concentrations of nitrogen. So probably in this situation, a cyanobacteria, which is a very efficient nitrogen utilizing mechanism, will outcompete the other taxa and will show a high rate of growth. On the other hand, dissolved inorganic phosphate, in a condition where there is enough availability of phosphate, obviously in this case also it will allow diversification of phytoplankton population. But when phytoplankton or becomes exposed to very low concentrations of phosphate under phosphate limited conditions, so in this case those taxa which have the capacity to form polyphosphate bodies or those which have the enzyme alkaline phosphatase, they would probably adapt better to this situation to utilize low levels of phosphate. So in those conditions also there will be selective preference for some taxa and that will outcompete the other taxa. So in these kind of limited condition, nutrient limited conditions, the possibility of a bloom event becomes much higher. So nutrient limitation is not a preferred condition for any aquatic ecosystem. So nutrient limited condition may actually lead to conditions of phytoplankton blooms. The other important parameter to regulate a phytoplankton population is the location of pycnocline. So the term pycno means colored. So a gradient of colored particles actually forms the pycnocline. So it can be of biological origin or it can be of non-biological origin, that is sediments. So if the amount of sediment load is low, so it will form a shallow pycnocline, and then light availability will be more and the producer community will be able to grow and reproduce, and that will make the ecosystem more stable. Whereas if the amount of suspended particulate matter or particulate matter of non-biological origin becomes high, so it will form a deep and thick pycnocline. So as a result of this thick pycnocline, light availability will be severely hampered, which is measured by the light attenuation coefficient. So higher the light attenuation coefficient, lesser will be the availability of light and higher will be the availability of suspended particulate matter. So there is a positive correlation between suspended particulate matter and light attenuation, whereas there is a negative correlation between suspended particulate matter and light availability in the water column. So as SPM and light attenuation coefficient increases, so light availability in the water column decreases, and that would lead to less production by the phytoplankton community, and that will make the ecosystem unstable. Finally, the other parameter or the important parameter is the temperature. So obviously as there is formation of photic zone and aphotic zone, so obviously there will be a thermocline as well. The region of water column which is exposed to light will have a higher temperature, and that region which is not exposed to light will have a lower temperature, and that will create a gradient called the thermocline. Now most phytoplankton populations doesn't have the heat shock proteins. So most of the phytoplanktons are not adapted to changes in temperature or high changes in temperature. Most phytoplanktons are not thermophilic. So obviously there is an optimum range for the phytoplankton populations to grow. If the temperature range goes below or beyond this range, then obviously phytoplankton populations cannot proliferate. So there is a direct relationship between the light availability and the thermocline of a particular area. So better the light availability, better will be the thermocline, and here also since the surface water will be more heated, the metalimnion or the middle part of the thermocline will be less heated, and so the thermocline or the middle part of the thermocline corresponding to the metalimnion zone of light availability will be the preferred place for the phytoplanktons to grow. That is why the metalimnion region is considered as the zone of chlorophyll maxima, not only with respect to light availability but with respect to temperature as well. So moving on, these are some of the images of bloom events in different parts of the world. Now the word bloom can be applied particularly in conditions where the cell count of a single taxa reaches more than 20,000 cells per ml. Now obviously in such a high cell count on the surface of the water, light availability for the submerged aquatic plants will be minimal. Now since a byproduct of photosynthesis by this submerged aquatic plant is oxygen, so non-availability of light due to this bloom event will lead to non-production of oxygen, and that will lead to subsequent hypoxia of the water column. That may result in fish kills. The other important aspect or the other negative aspect of bloom events is the aspect of toxin production by different phytoplankton forms. Now many of these toxins are neurotoxic and some are hepatotoxin. Now in case of hepatotoxin, there is inhibition of different phosphatase enzymes and enzyme like phospholipase A. Now, since the cell membrane is made up of proteins and lipids, and if phospholipase A is inhibited, so obviously the membrane becomes more and more leaky. So those plant forms or those animal forms which are exposed to these hepatotoxins produced by different dinoflagellates will obviously die, and that is a condition known as diuretic shellfish poisoning. Then there is another condition called ciguatera shellfish poisoning. Here also this kind of hepatotoxins are produced of different chemical nature, and they disrupt the voltage gated channel or the calcium channels that causes pH homeostasis and such changes, and thereby leads to fish kills. The other type of toxin are the neurotoxins, which are produced by taxa like gymnodinium. Now, these neurotoxins basically disrupts the development of action potential. So, they are deposited at the synapse region and they disrupt the transmission of signals between the neurons. This leads to paralysis, and the phenomena is also known as paralytic shellfish poisoning. So here are some images. As we can see that this nuisance dinoflagellate called Canaria brevis, they produces different lethal toxins, and when they are consumed by the fish, it causes huge fish kills. So fish kills can be both due to hypoxia as well as due to these neurotoxins. Then there is another taxa called Gambier discus toxicus. Now this Gambier discus toxicus actually forms different kind of cysts, and they remain attached to the kelp forest or to macroscopic alga like ascophyllum. Now when this ascophyllum is fed upon by different fish, they also consume this cyst of Gambier discus. Now once this cyst gets inside the system, then they starts functioning and they start producing this toxin. So that also leads to fish kills. Now in the lower panel we give a picture where we can see that a humpback whale has also died by consuming fish which were laden with these toxins. Now since there is this biomagnification in this humpback whale, so that also has led to death of this humpback whale. So we can understand that even though phytoplanktons are important, but bloom events, especially for those taxa which produces toxins or can lead to hypoxia, is a deterrent for aquatic ecosystems as a whole. So finally we come to the conclusion that as the earth is getting heated up, so obviously precipitation events are getting more and more localized. In some places we are getting flood events, whereas in other parts it is getting warmer and warmer. So, moreover, we know that global warming is a well accepted phenomena at present. So we all know that global warming is happening. So as global warming keeps on happening, so it is melting away the freshwater resources, and that is adding up to the volume of the marine water. Now as marine water expands, so it enhances salination, and it is inundating new and new areas which were previously not submerged. Now moreover, as the earth heats up, so the process of evaporation is enhanced, and that leads to quicker salination. Now all these factors play hand to hand, and thereby makes the habitat more and more saline. So in such a situation, halotolerant forms will have a preference with respect to available conditions as compared to stenohaline taxa. So obviously there will be a shift in the phytoplankton community. The other aspect is that as the water column on the surface is getting more and more heated up, the gap between the lowest part of the water column and the topmost part of the water column is getting wider with respect to temperature. Now, this phenomena is called stratification. Now, as the water column gets more and more stratified, so the solubility of oxygen changes rapidly over small depths. And such changes in oxygen often may cause depletion of oxygen and development of dead zones. And obviously, we all know that in dead zones, life forms cannot survive. Along with it, there is this phenomena of ocean acidification as well, which we have already discussed. So as there is a shift in the bicarbonate to carbonate pool, so obviously some of the phytoplankton communities which are dependent on the carbonate pool will be hampered. So the other aspect that is also ruining the ecosystem functioning is the aspect of eutrophication brought about by anthropogenic pressure. So when all these factors play hand to hand, the foremost parameter that is expected to get disturbed is the phytoplankton community. So if we observe the phytoplankton community closely, then the changes in phytoplankton community can be an ideal proxy to understand the signals of climate change. So it's important that phytoplankton gets monitored at regular interval. So I would request the scientific community and those who are listening to this lecture to get more and more coherent with phytoplanktons, because they are actually one of the important parameters of natural ecosystem. We can do more and more scientific research on different aspects of science, but then we need to conserve our ecosystems first. If we have our ecosystems, then we are there. If we don't have our ecosystem, then obviously our races and populations will be limited. So in this situation, study of phytoplankton is important, and it will continue to gain more and more importance in the coming days. So I will request the audience to kindly be observant on phytoplankton. And yes, this phytoplankton also can be a very interesting and important in the area of both study and research. So at the end, I would like to thank Probir Chatterjee Research Foundation for giving me this opportunity to give this lecture for their upcoming YouTube channel. I hope this will be an enriching lecture for the audience, especially those students for which I have given this lecture. I again thank the Probir Chatterjee Research Foundation from the bottom of my heart for giving me this opportunity. Thank you.